All right, hello. So I have tried my very, very best to compel pretty much every single learning objective in every single practice test question that I think will be possibly useful for this exam because obviously I make a YouTube video like this personally to help my own studying because I'm trying to take the test as well. But I mean, if any of you watch it, say if you're taking this class a year from now and you just want a quick rundown, you can put the video on 2x replay and you can get a gist of everything you would need to know for the course. But this is going to be a final exam review of me teaching this entire course basically to myself off of this 150 page document that I have that I've been studying. Me talking about it obviously helps myself. So I'm going to go ahead and get into this video and start talking about it. Okay, lesson one of 140. Holy cow. Okay. Biological diversity is the study of variation among living organisms, hence the words biological. Yeah, that makes sense. Biodiversity. A dilution host refers to an organism that reduces the likelihood of a disease transmission within an ecosystem by diluting the impact of more competent hosts. Okay, so that makes sense. So, say for example, like Lyme disease, where you have say a tick that gives a disease, or and you have a dilution host, such as a possum, that possums can get these ticks and like the diseases don't affect them as much. So if you have a lot of possums in an environment, then those ticks get on the possums instead, and the possums stay healthy. And in turn, they don't get on humans. So less humans get Lyme disease. So if in an environment you have a dilution host, say like go extinct, that could be say harmful for a human population. You would see more disease transmission. And biodiversity is pretty much how how different organisms in a community all interact with each other in all these different special types of ways and all these ways you can analyze them and whatnot. Just gonna go through the important stuff. All right, so when you're doing an experiment, you have your independent variable, a dependent variable, a control group, an experimental unit, or yeah, experiment unit. So our independent variable is the variable that is manipulated or changed, and the dependent variable is how we measure that change. So basically, if I were to do an experiment that says how many hours of sunlight per day affects the growth of plants, we could have one group of plants that we leave in the dark. We could have one group of plants that's outside that say gets sunlight 12 hours a day. And we have another group of plant that stays in a closet and gets sunlight all hours of the day. Our independent variable would be hours of sunlight, and our dependent variable would be like plant growth. So independent variable is just a thing, where dependent variable is like measurable outcome. And our experimental group is a group that receives a treatment. So experimental group would be um, group that receives light, or a control group would be like a group that doesn't receive anything. I guess it's just outside. And our experimental unit would just be like one plant. So that'd be one unit of whatever we're testing. Ecosystem services. So if there's a question about something does X, Y, and Z, what type of service is it? We have supporting, regulating, provisioning, and cultural. Supporting ecosystem services have basically everything to do with like nutrient, nutrient, nutrient cycling and in like soil and whatnot. So if you think of like anything with like the nitrogen cycle, anything that has to do with soil or like nutrients in the ground, is supporting ecosystems. And regulating is like climate control, food control, disease control. Provisioning is food, water, fuel, medicine, basically any use you can get out of it. And cultural is like anything that would be of use to humans. So, say you can use this area for recreation or whatnot, whatever it would be. Yeah, so supporting is nutrient cycling, and regulating has everything to do with just maintaining an ecosystem and making sure it stays healthy. Provisioning is everything that you basically would need to survive, and cultural is what would be of use to humans. Yeah, so there's a question here. By minimizing otter illness, which ecosystem service and wetlands are we providing? So basically, we just said how these animals can serve as dilution host. So if there's more dilution host, then that means that it's going to be better for humans. So if we minimize, I think it's saying by minimizing otter illness, which ecosystem? Okay, so this has to do with, say, like disease transmission. So we just said that disease transmission would be, or like sickness would be provisioning, because you see, like, no, not provisioning, regulating, because disease regulation. Provisioning would be medicine. So you see down here, it's correct answer would be regulating services, because it's like regulating that environment, making sure it's staying where it is. And the explanation is regulating services include benefits that result from the regulation of ecosystem processes, including Earth's climate, water purification, and food control. That's a good explanation. So I just quizzing myself again. Supporting has everything to do with nutrient cycling and soil formation. Provisioning, that is like our food, water, and our medicine. Our regulating is flood control, climate control, disease control, and cultural services is like everything that would be of use to humans. So this is our way of measuring biodiversity. So say if we went to some new island and we wanted to measure the biodiversity of the species on the island. We could look at the species composition, the species richness, and the species evenness. So basically, our species composition is just the number of species that are present. So if we had 10 different species, we would say that there are, that's not correct. The number of species is species richness. So if we were looking at the species richness, we would want to see how many species we have. Species composition is just what like the name of what you would have. So I mean, like, we went to an island and there was a squirrel, a duck, and a pig. Our species composition would just be squirrel, duck, pig. If you were asked what's the species composition, you say squirrel, duck, pig, versus species richness, you just say three, because there's three species. And our species evenness is like the proportion of how the species are close together. So, say for example, we had 10 ducks, three pigs, and five squirrels. That would be the species richness. So if we have two separate islands, they could have the exact same species richness and have the exact same number of species, but the evenness could be completely different. That's a note. Morphological species are visibly different. Biological species can only produce variable offsprings with members of their own species, but may be hard to distinguish visibly. If that makes sense. If two organisms are able to reproduce with each other, then they're likely the same species. Just like that makes sense. And this is our way of analyzing biodiversity with our alpha, beta, and gamma diversity. Oh yeah, an endemic species is one that's only found in one habitat or area. So if you're asked what's an endemic species, you could say penguins because they're only found in Antarctica. They're not really found any other place of the world. So our alpha diversity is the total number of organisms in an area. So with that previous example, if we had an island that had squirrels, pigs, and ducks, the alpha diversity in the area would be three because there's three species. And we look at beta and gamma diversity when we're comparing two separate areas. So say we have, or you just look at this example I have right here on the screen. So if we're looking at alpha diversity for site A, it's 7 because we have seven separate. Look, you could say species, but shapes are basically. And then for B, the alpha diversity is 5. For C, the alpha diversity is 7. And we look at beta when we're comparing different species. So our beta diversity is basically the the number I put it here as the number of organisms that are unique to both groups combined. I think I can probably find a better definition of that. But I'm pretty sure the best definition of beta diversity is the total number of species between the two groups that they don't share. So say for example, area A has squirrel, pig, and duck, and area B has squirrel, pig, duck, deer. Even though there's 4 species in area B and there's 3 species in area A, you would only say the beta diversity is 1 because the deer is the only organism that they don't share. Versus you could have island A that has animal A, B, and C, and you could have um, area B that has animal X, Y, and Z. Then their beta diversity would be 6 because there's 6 organisms that are not shared between the two. That's the best way I can explain it to make sense. I think I have a picture in here that I put that explains it way better because it was difficult for me to understand at first. Yeah, right here. The beta diversity is the total amount of species between the two groups that the two groups do not share, and our gamma diversity is the total amount of species in the two groups, not counting the repeats. So, say like we have area A that has pig, duck, and squirrel, and area B also has pig, duck, and squirrel. We would say that the gamma diversity between the two areas is 3 because there's pig, duck, and squirrel, but we're not going to count the repeats. Versus if there was pig, duck, and squirrel over here, and pig, duck, squirrel, deer over here, our gamma diversity would be 4 because there are 4 species, not counting repeats. That is a much better way that I wrote it as. That makes sense to me. In our Shannon Wiener, and that's a funny name, Wiener Index, it combines our species richness and our evenness to measure the biodiversity of an environment. So it's pretty straightforward. I need to make sure my calculator is charged now. But basically, if you're asked to calculate the Shannon Wiener Index, you basically take, how did I write it here? Once again, I have a way better example down here that I'll talk about. the Shannon Wiener index. I'll get my calculator just to make sure it's charged. Okay, so if we're asked to calculate the H value, right, which is what the Shannon Wiener index is, which is a measure of alpha, a measure of biodiversity in the environment, right? So what we do is we look at how many number of organisms or individuals are in that environment. So if you look at the example on the left here on the top for Huggins Island, it's 75. And it looks like we have 40 of individual of species A, 20 of species B, 15 of species C. So what we do is we take the log of that number of species for species A, and then we divide that by the total number of species, so 75. We multiply that by 3.22 because it's a constant, and then we once again multiply by that same fraction again. And then we do that exact same thing for every single number of species that there is. So we would do log of 20 divided by 75 multiplied by 3.22 multiplied by that fraction again, log of 15 divided by 75 multiplied by 3.22. And then you basically just sum all of those values together. And then when you get it in your calculator, it's going to be a negative number, but you just flip it to a positive. So it's pretty straightforward. That's how you would calculate the H value. And once again, that is a measure of that takes into account the species richness and species evenness of an area. Yeah, I put a note in here. Difference in H value is the difference in species richness and evenness. So if we had two different areas that had different H values, we could explain that by being okay. The two areas have different species and two different species richness and evennesses. Which sites need continuing sampling? So if you get a map that says which sites need continued sampling, basically this curve basically represents how many species you found, and this is like number of species found versus trials. So you can see if we start in the first couple number of trials, right, we see like an exponential kind of growth here. But as we do more and more sampling of an environment, you're going to see that curve starting to flatten out because that means we've kind of found everything. So each line represents a different area that's being sampled, right? So it looks like for example, the blue area. It looks like with the blue area, we're still sampling and we're still finding new organisms because it's still a positive linear slope. Same thing with the pink area. It looks like purple. Out of all of these, purple is the only one that may looks like it's at a plateau and you don't really need to sample it anymore. But the question is, which sites need continued sampling? I think I put in here blue and pink, but I mean, yeah, blue and pink is the best possible option. But it looks like, yeah, yeah, blue and pink would need continuing sampling because as you keep sampling, you're still finding more and more species. So you would have to keep looking until basically the line plateaus, which may take thousands of trials, but you never know. All right, what factors, what external factors of the Earth basically affect biodiversity? So we have our orbit, we have the Earth's tilt, also known as obliquity. So we have the green planet here, which is the Earth, and we have this red planet over here, which is a planet that has a perfect circular orbit that has no tilt. And the difference between these two planets is, unlike the Earth, it would have the exact same climate in all areas because there's no tilt. Like it's getting the exact same amount of sunlight in all areas. It would have a perfect 12-hour day and 12-hour night because it doesn't have it has a perfect orbit. around the sun, and there will be no seasons because there's no Earth's tilt. Versus with the Earth, it's like slightly tilted. So in the northern hemisphere, when um, during the summer, when the northern hemisphere is tilted closer to the sun, we have summer, and in the winter, when it's tilted farther away from the sun in the northern hemisphere, we have winter. So all of these factors, external factors, kind of influence like why we have our seasons basically, and why we have different night and day hours, and why we have different climates in different parts of the world. Yeah, what I hear, I put Antarctica gets very little direct sunlight. That is why it's so cold. Antarctica is also a desert because it gets little precipitation. Yeah, that's true. I have a map about that. That's interesting. Solar radiation plus tilt, yep, equals our seasons. Warm air rises, cold air falls. Talk about the Hadley cells in a second. Okay, so this is our Hadley cells. So our Hadley cells basically explain how pressure circulates in the environment, leading to different biomes and regions. Like, why at the equator do we have really tropical rainforest, and why specifically at 30 degrees north and south of the equator do we have really, really dry desert terrains? Like, if you look at this map here, you see the little red line here. We have some parts of South America where there's a lot of precipitation, a lot of tropical rainforest. So you look at 30 degrees north and south here, you see like the southwest United States, parts of Mexico where it's a big desert, right? So basically, right at the equator, there was always basically direct sunlight hitting the earth at all times. So what happens is this direct sunlight is coming into the earth, but warm air rises. So as it comes down, it's coming back up. And warm air rises, it expands, and then it causes rain. So that's why we get a lot of precipitation at the center of the earth, leading to a lot of tropical rainforest, and that's why it's warm all the time. But keep in mind, even while it's still raining and that cool air is expanding and causing rain at the equator, there's still constant light still being pushed down. So this light, or this, is going to kind of push the cool dry air to the side, and it's basically going to blow over at specifically 30 degrees north and 30 degrees south of the equator. And that's why we have these kind of we have these high pressure, really just dry environments at 30 degrees north and 30 degrees south of the equator. And then it goes back around too. Like, you have the Hadley cell that's 30 degrees. We have the Ferrel cell, and then we have the polar cell. That's how I mentioned earlier that Antarctica is basically a desert, because if you look at it, it's on the part of the cell basically that gets a lot of dry air, basically a cool dry air. Like, there's little precipitation and never rains. So it's basically a desert. It's a tundra, but it's basically a desert. Yeah, so I have a map here of biome distribution, which basically compares annual precipitation to annual temperature. And as you can see, when we have high temperature and high precipitation, we get tropical rainforest. And when we get um, when we have really low temperature and low precipitation, we get like a tundra. Think of like Antarctica. We have all these little areas in between. Like in North Carolina, we're probably like a temperate forest, I would say. That's probably a good estimate. GPP, gross primary productivity, is the total photosynthetic photosynthetic production in an ecosystem. Net primary productivity (NPP) is GDP minus energy used by primary. Yeah, I have an equation for that. How does CO2 enter and exit the atmosphere on our planet? It enters via cellular respiration. So um, plants do photosynthesis, right? They also do cellular respiration. Fossil fuel and exits during photosynthesis, and a lot of CO2 is actually absorbed by oceans. So when plants do cellular respiration, they're releasing CO2 in the environment, right? And when plants are doing photosynthesis, they're they're using that CO2 basically. And if you look at your equation here, it's middle. You probably learned in middle school biology. We see that in cellular respiration, plants use basically sugar and oxygen to create um, as they generate ATP, basically, so they can live. And as a byproduct, they release CO2 and H2O. And photosynthesis is basically plants use light, CO2, and H2O to make glucose and oxygen. So if we were trying to look at which months have the highest CO2 concentration, we would want to look at during what months are plants doing the most cellular respiration, right? So during the summer months, a lot of plants are doing photosynthesis, right? Like they're getting their leaves, everything's green. But in the winter, there's a lot more cellular respiration. So if we wanted to estimate like what months of the year would have the highest CO2 concentration, it would be the winter. And as you can see here with number three, I put our highest months of CO2 concentration in the northern hemisphere would be the winter, and during the lowest, it would be the summer because RS means respiration, and then PS is photosynthesis, which is kind of what I just explained. Again, Antarctica has literally no variable CO2 concentrations at all because it's literally just a desert. There's no plants, there's no trees, there's no humans. Literally nothing happens there. So it never really varies there. Whereas you have some parts of the northern hemisphere that vary, and there's CO2 concentrations like significantly throughout the year. Ooh, blowing up my phone. I'm sorry. Give me a second. I'm gonna put my phone on silent. Sorry. All right. So external forces of climate is we call it the Milankovitch forcing or factors, which is basically like orbit, Earth tilt. It's known as the Milankovitch factors. Whoever Milankovitch was, he must have been an important guy to have something like that named after you. Greenhouse gases. This basically just shows how CO2 concentrations have basically increased over the past couple hundreds of years. So we look at years before present zero. So today, our CO2 concentration is like around 350 versus 1,000 years ago, it was like 280. So climate change is definitely real. So there's a lot more CO2 in the atmosphere because basically what happened is due to the Earth's greenhouse effect, which I mean you typically learn about in middle school. The greenhouse gases in the atmosphere are responsible for Earth's environment. So basically, sunlight comes in, and the greenhouse gases in the environment absorb basically some of that heat from the sun, which allows us to have a warm environment, basically, instead of a freezing cold environment. So CO2 in specific um, absorbs this heat. So if we're thinking about increasing CO2 concentration. We're releasing more and more and more into the air, right? That's going to be more CO2 to absorb more heat from the sun. And in turn, that's going to make temperatures go up. So, I mean, that's how it works. And you look at some planets, I think it's Venus. I might be wrong about that one, but there's one planet that's in the Milky Way that I remember. There might be an example in this PowerPoint. It's like the environment is like 80% CO2. Earth, I think Earth's is only like was like 3 or 4%. Let me Google how much of atmosphere is CO2. 0.0%. So carbon dioxide makes up 0.04% of the world's atmosphere, not 0.4% or 4%, but 0.04%. Oh no, so it's not 3 or 4%. It's 0.04%. Really, really small. So, for example, if you look at a planet like Venus that is 80% of their atmosphere is CO2, it's going to explain why it's so hot. Let me fact check that. Oh no, Venus is 96% carbon dioxide. It's more than I thought. That's why it's so hot. It's 96% carbon dioxide. Oh my goodness. Um, how hot is Venus? 864 degrees Fahrenheit. Yep. It's pretty hot. Okay. Greenhouse gases. Yeah, yeah, yeah. They're rising. We know. All right. So we have our nitrogen cycle. So um, the Earth's atmosphere is 80% nitrogen, and a lot of the nitrogen in the air is basically useless to plant life because it's N2 gas. It's not N3 or ammonium or nitrate. So it's useless. But we have this process called the nitrogen cycle where microbes basically in the ground, like microbacteria or little bacteria inside the soil, basically get hold of this nitrogen and they basically break it down, and plants use this nitrogen basically as energy to grow. So microbes, short for microorganisms, are tiny, very microscopic entities that are too small to be seen with the naked eye. They include a diverse array of organisms such as bacteria, viruses, fungi, protista. Yeah. So all these basic type of bacteria are in the soil doing work in this nitrogen cycle. So our cycle is we have N2 gas in the atmosphere, and I might have some better terms. So N2 in the atmosphere basically gets into the ground as ammonium. And plants can uptake this ammonium or it can be immobilized and just stay in the soil. I have some better terms here. So we have fixation, immobilization, uptake, mineralization, nitrification, denitrification, and leaching. So fixation, this is the process where nitrogen gas from the atmosphere is converted into ammonia by bacteria. So fixation is how does the N2 in the air get into the ground as ammonia, which is useful to plants. Immobilization, this requires the conversion of inorganic nitrogen forms into organic nitrogen forms by microbes and plants, making it unavailable for use by most plants. So you can see in this map here, mobilization is kind of where it comes to the side here, and it's useless. Can't really use it. And then uptake is this is the absorption of nitrogen by plants from the soil, where it is used to form amino acids, proteins, and other essential um, organic compounds. So plants uptake this ammonium or nitrate and they basically use it to grow. So if you could think of there being more nitrogen in the soil, you would see increased growth in a plant population, right? Or you'd see excess growth. Mineralization, also known as ammonification, this process involves the conversion of organic matter back into inorganic forms, such as ammonia, which can be used by plants. I think mineralification, yeah. If you look at soil organic nitrogen pool, that just basically explains how when things die or animals poop, that goes into the ground and it gets broken down by those microbes in the soil. And then that is once again absorbed into the nitrogen cycle. So that's another source of how um, nitrogen gets in the soil. So mineralization, also known as ammonification, this process involves the conversion. So this is how mineralization is how nitrogen gets turned into ammonia. Is there a picture of that mineralization? So yeah, this mineralization would be how, say, feces would get turned into ammonium, which is available for, say, uptake by a plant. Because uptake is usually in the form NH4 plus and nitrate NO3 minus. So nitrification. This two-step process involves the conversion of ammonium into nitrate and then nitrite and then into nitrate by specialized bacteria. So if you think of nitrification, it just thinks of how is ammonia, which is NH3, going to go into nitrate and nitrite, which is then absorbed by plants. Denitrification. This is the process of how nitrates get turned back into nitrogen gas and go back into the environment. In leaching, so say if there's like a big rain, if there's a big flood and a lot of nitrogen just got taken away from soil, we would call that leaching. So I think the definition I put here is this is the process where nitrate is removed from the soil with drainage water. It is a concern because it can deprive plants of nitrogen and contaminate water sources. That's true. So, in summary, we have fixation, immobilization, mineralization goes into ammonium, nitrification turns ammonium into nitrate and nitrite, and ammonium and nitrite is available for uptake and use by plants, which promote growth. And then denitrification is when nitrate and nitrite go back into the atmospheres and leaching is when nitrogen gets taken away, basically. I think I put a note in here is that heat from lightning can turn into ammonium, which I think is interesting. All right, so dead zones. So I also mentioned how nitrogen promotes growth, but if say an area has too much nitrogen, that can be extremely harmful because say there's excess nitrogen, say in a pond, and then due to that excess nitrogen, you have a bunch of algae that blooms at the bottom of the pond. Now, basically, that algae down there requires oxygen to basically survive. So now all of a sudden you have increased algae population at the bottom of a pond. It's basically absorbing all of the oxygen, which makes the water hypoxic, basically low on oxygen. And then it basically just kills off all of the other organisms in the lake due to lack of oxygen, basically. And then the pond becomes known as what is a dead zone. So that's why when we talk about if, say, a farmer uses too much nitrogen and then all that nitrogen gets taken away into an ocean by a flood, then that can be extremely harmful for the ocean population. And a problem with that now is all of these farms in the Midwest United States that use a lot of, say, like fertilizer with nitrogen, all of that gets washed into the Gulf of Mexico, right? So now you have this problem where you have all this excess nitrogen being ran into the ocean, and that's causing excess plant growth, which makes it actually more difficult for organisms to live in the ocean, which has a more harmful effect than good. So I think so. Order the steps: fertilizer, nitrates and nitrites move into the ocean, promote algae growth. Most of the algae is uneaten and dies. Can also block light too. Decomposition of algae removes O2 from water, and then lack of oxygen, it becomes a dead zone basically. Yeah. A carbon sink is a forest slash ocean. Anything that absorbs a lot of carbon is a carbon sink. And detrivores are decomposers. Autotrophs create their own food. Heterotrophs consume food. Carnivores eat meat. Herbivores eat plants. Yeah, that's good to know. All right, this is getting more into what was on the second midterm. That was all the first midterm, basically. All right, what is this talking about? This talking into human evolution, kinda. So this is a phlebotomy, not phlebotomy, phylogeny chart. So a lot of people think that humans evolved from monkeys. That's not necessarily true because monkeys and humans are two completely, totally distinct species. But if we look at this phylogeny here, each little branch here that we have is a common ancestor. So, say starting millions of years ago, there was this common ancestor right here. And then among these branches here, there's evolution occurring. But each of these little branches here are common ancestors. So, if we were asked what is the last common ancestor that humans had with lemurs, it would be this. Because we don't know what this common ancestor is. It could have been something completely different, but we know that we had a common ancestor with, say, monkeys, but we're not monkeys because monkeys and humans are two totally different species, and they evolved into monkeys along this path, just like we became modern humans, right? So humans didn't evolve from monkeys or gorillas. We all evolved from the same common ancestor. So if we were asked, um, what common ancestor do humans and Neanderthals have most in common? It would be this one right here. Or we would be asked, um, are humans more closely related to um, chimps or bonobos? The answer would be they're both equally related because they come from the same common ancestor, right? Humans are still evolving. Um, every species that is alive today has been evolving for the same amount of time as humans have. That's interesting. Humans are still part of the tree of life. They are not above it or below it. That's very true. Humans are Homo sapiens. They are a species, just like everything else is a species. It's interesting to really think about because your whole life you grow up thinking humans rule the entire world. Which I mean, it's kind of true, but I mean, you still have to understand that we are our own species known as Homo sapiens, which is interesting. What are these different hypotheses for how the world was created? So we have our young Earth creationism, which is God created the world. Hypothesis two is that Lamarck's hypothesis is that individuals adapted to change, which is not true. If I'm short, I can't just decide to grow taller in my life because I need to be taller for my job. That's not how it works. Hypothesis three is Darwinian evolution, which is one that everyone should learn about in middle school. And that's kind of what we learn about in this class in specific. There's another thing. I talked about GBP and NPP. So gross primary productivity is equal to respiration plus the net primary productivity. And obviously you can switch those equations around. Like, for example, GPP minus respiration is equal to NPP. So gross primary productivity is how much energy goes into the environment. Respiration is how much is used by plants for respiration. And net primary productivity is how much is left after respiration for organisms to use. So if the question was 800 joules go into an environment and 100 are used for respiration, what is the net primary productivity? The answer would be 700 because 800 comes in, 100 is used for respiration, and 700 is left, right? Or you could get another question that would be plants use 60 joules for respiration, and now there's only 600 left for everything else. What's the GPP? We would know it would be 660, right? Because that's how much originally came in before respiration. Natural selection and mutation. So high fitness, like species fitness, basically just refers to how good a species is at reproducing, basically. Like if a species is really fit, it's really good at reproducing and staying alive. Versus some species that have really low um, fitness are going to go extinct. Look at humans. Humans are incredibly good at reproducing um, in comparison to most other species. I mean, like 8 billion. I mean, humans are basically invasive because there's nothing to stop population growth. So yes, humans have very high um, fitness. And if you look at this map here, we have this green bug looks like it has 2 kids. This brown one has 4, and obviously it ends up passing its genes on more and more and more. Presence of variation is key to evolution. Mutations allow the production of new alleles, and sexual reproduction causes variation. So yeah, we have sexual reproduction, asexual reproduction. Sexual reproduction is slower, but it allows there to be genetic variation, right? Because you have the crossing over of chromosomes and mitosis and meiosis. And then asexual reproduction is very quick, but there's very low genetic variation. So if you had a population that could reproduce sexually and a population that could reproduce asexually, um, that asexual population is going to be a lot more vulnerable to, say, going extinct if there was, say, a disease because it only has one combination of alleles that so if that combination of alleles is affected, that entire population is dying. Versus if you have a population that can reproduce sexually, all of those alleles are different. So some people are going to be affected and some people are going to be completely fine. A mutation is a random and permanent change to a DNA sequence that literally just happens randomly. So, I mean, if we're looking at why alleles change, it could be due to a mutation, which is completely random. It could be due to selective breeding, which means that individuals with the most selective alleles reproduce while the ones that don't reproduce, and that's why you see that allele change over time. And then mutation, selection. There's another one in there. Mutations are random. It does not give a species what it needs. If a species gets an unfavorable mutation, it may go extinct. Mutations often worked in the opposite direction of natural selection. So, meaning natural selection is a very slow, happens for a kind of a specific reason. Like, for example, if birds with pink feathers are better at surviving than birds with blue feathers over a long period of time, the birds with the pink feathers are just going to basically dominate the entire population. Versus mutations just happen randomly, immediately. So they kind of go against natural selection. So our three things must be present for natural selection to occur. Variation, meaning that there must be differences between traits. Number two, these traits must be heritable. And number three, there must be a differential reproductive success, right? So variation, heritability, differential reproductive success. More successful traits will increase in frequency in a population over less successful rates. That's very true. Mutations, a change in a gene's DNA sequence to produce something different. It creates a permanent change to the DNA sequence. Yes. Random mutation. Resistance mutations occur after the phage is present. Every cell will not have an equal probability of resisting to mutation, mutating to resistance. Induced mutation. Resistance mutations occur before the phage is present. Every cell will have an equal probability of mutating to resistance. Sickle cell. Population genetics describes the transmission of alleles in a population. How do we know if evolution is happening? We use our Hardy-Weinberg equation to basically check how our, so we have, you learn about this in very basic biology, but you have, say, heterozygous dominant population, which or individual that would have allele big A, and you have a heterozygous recessive, which would have alleles small a, and then you have a homozygous, which would be big A, little a. So our Hardy-Weinberg equation lets us know the frequency of alleles and genotypes in a population. And we use our equation p squared 2pq and q squared. And p squared plus 2pq plus q squared is equal to 1. So when we do our calculations, we use our Hardy-Weinberg, which is a null model. Basically, it means it's not really accounting for any changes, if that makes sense. But if we were given the list of alleles in a population and we tried to also calculate what it should be, if we know that what it should be isn't equal to what we currently have, we know evolution is occurring. Versus, this is the best way I could say it, of if observed allele frequency is equal to expected allele frequency, then evolution is not occurring. But if the observed allele frequency is different from the expected allele frequency, then we know that evolution is occurring because something is changing in the environment. Yeah, so if we look at this gene pool, it looks like we have 5 individuals and we have 10 alleles. So it looks like we have 6 dominant alleles, 4 recessive alleles. So we would say our P, which is our dominant, is 0.6. That's the frequency of it. And our Q, which is the frequency of the little allele, is 0.4. So P plus Q equals 1. And then once again, if we were to look at, it's pretty straightforward. P squared is just P times P. And you have 2PQ and Q squared. All of these help us calculate our allele and genotype frequency. And the equations are pretty straightforward. P squared plus 2PQ plus Q squared equals 1. And at the top there, just that big A or the little a, that refers to our allele frequency versus big A, or big A, little a. That refers to our genotype frequency. So we have genotypes and then allele frequencies. And our Harvey Weinberg is a null model. Basically, it doesn't account for sexual selection, mutation, migration. It's basically just assuming that the population size is huge and mating is random. So I'm not saying that's impractical, but it's not a real representative model of the real world, if that makes sense. It's just used to test things. And yeah, our Mendelian genetics is the transmission of alleles from parents to offspring. That's what it explains. Yeah, so heterozygous pair, homozygous pair. Observed not equal to expected in Harvey Weinberg means evolution has occurred. Yeah. So there I just explained genotype. Our genotype is basically our genotype is basically what the letters are, basically. So our genotype would be we have um, homozygous dominant, heterozygous, or homozygous respective, homozygous recessive. And our phenotype is what we observe. So say like if if having one dominant allele makes you have brown eyes, and having being homozygous respective makes you have blue eyes, we would say that there's only two phenotypes, blue eyes and brown eyes, even though there's three genotypes. This is basic biology stuff. I'm just reviewing it for my sake. Sexual reproduction. Yeah, I basically talked about the benefits of asexual reproduction. It's very quick, it's very easy, but also it's not good in terms of it's very vulnerable because obviously if there's a bad mutation or a bad disease that can literally wipe out the entire population versus sexual reproduction, um, it's slower but it's more resistant to um, like random mutations or random changes or random diseases, if that makes sense. We're at sexual reproduction. So basically, the gist of it is sperm is cheap and plentiful. It's a very easy for a male to produce sperm. Males basically produce sperm their entire life, versus eggs for females are um, they're not as cheap and plentiful. So in most um, species, right, the goal of the male typically is to just try to basically reproduce with as many females as possible. So the male's objective is to reproduce with as many people as possible, versus the female's objective is to try to get the most desirable trait, right? Because there's another thing in here that explains that better. Yeah, so um, sperm's fitness is limited by the ability to attract mates, so they maximize offspring number, versus egg fitness is limited by an ability to gain the resources required to produce eggs and rear young, because there's a finite number of eggs in females, right? I mean, even like if you look at humans, I mean, like the average woman can start producing as soon as they basically hit puberty, and then they go to menopause, versus men can pretty much reproduce their entire life, right? So female's objective is to maximize offspring quality, while male objective is to maximize offspring number. We look at this map here that says duration of copopulation. Well, I think it's just duration of sex, right? Versus a chart of body size length, so of nuptial prey. So some males, like bugs, right? They'll literally give a gift of a prey to a female they're trying to attract. And basically, this chart here basically shows you that the bigger the prey is, basically the more sperm that the male um, bug ends up giving away, right? So it's, and then also, what does this one say? Yeah, that makes sense. Like, as females are, even in a biological sense, are always looking for, trying to get the best possible, if that makes sense. Trying to maximize quality. And that's why you'll see in this example here, you have in certain species, you'll have most females in general reproduce, have very equal reproduction rates. Like the average female will reproduce versus if you look at male reproduction mates, you'll have a good majority of males that reproduce multiple times, and you also have another proportion of males that never reproduce at all. So males maximize offspring number, females maximize offspring quality. Genetic drift impacts small populations greater than large populations. So two populations are divided in half and blocked from seeing each other. So if we have two populations that are exactly the same and then they're divided in half, over time, due to genetic drift, natural selection, sexual selection, um, mutations, it may look like something like at the bottom over time. Because you got to understand, if there's two populations that are dropped in two different environments, certain species and certain traits might be able to develop better in one environment than another, if that makes sense. So natural selection, sexual selection, genetic drift, and mutations cause kind of populations to evolve over many different number of um, generations. Phylogeny, kind of what I explained earlier. If you have with the humans and the monkeys, we have our common ancestor. So phylogeny basically just explains how organisms relate to each other. And so if you look at here, humans and mice have this common ancestor here, and then among this part, humans have evolved, and among this part, mice have evolved. And each little break here is a common ancestor. So um, the dash part here is the shared history of mice and humans, and then the pink part here is the unique history of humans, and the blue part here is the unique history of mice that humans and mice don't share. A clade is a collection of branches and nodes that includes an ancestor and all of his descendants. A clade is monophyletic. Think of a phylogeny as a nested hierarchy of clades. So if you look at here, this pink part is a clade, right? You have all these different types of clades that are unique to each individual organism. Prokaryotes in fish are not clades, they are paraphyletic groups. Okay. Is the frog more closely related to the fish or the human? Is the frog more closely related to the fish or the human? Well, the frog's right here, and you can see that the most recent common ancestor it has with the fish is right here, and the most recent common ancestor that humans have with frogs is right here, which is X. So humans are actually, the frog is actually more closely related to the human than the fish because it has a more recent common ancestor with the human, which will be X. And what I put here, turtles are more closely related to birds than to mammals, right? That makes sense because this this phylogeny here just basically represents millions of years. So this just because it's evenly spread out here, for all we know, the distance between W and N might be 100 million years. So turtles are more closely related to birds than mammals because they share a more recent common ancestor. And just because there's a straight line here does not mean that evolution is not occurring. So for all we know that this common ancestor that everything comes from probably looked nothing like any of these organisms do now. They just all evolved among their own individual paths. And if we were to zoom into these individual paths, they would get more and more complicated. If that makes sense. In inflammogenies, the most parsimonious model is the one that requires the fewest number of evolutionary changes. So if you get a question that asks, this map has this, which is the most likely, what trait did the common ancestor most likely have? It's usually the one that causes the least number of mutations or changes, right? So if you look at this one, use parsimony to choose between the hypothesis. So if we look at here, lungfish, frogs, turtles, birds, and mammals, it's comparing limbs, eggs, and endothermy. So if we look at our mutations are all the different things that would have to happen for each of these to occur. And we're looking for the one that has the least number of transitions or mutations. That's the one we're always looking for. All right, population growth. When birth rate is higher than birth rate, population will grow, and vice versa. Exponential growth occurs in populations with continuous reproduction and overlapping generations. For example, humans, exponential growth due to antibiotics. There's literally nothing that has ever stopped humans for the past 100 years, pretty much. Due to antibiotics and safer births, nothing has really stopped humans over the past 100 years. That's why the population has gone from like a billion to 8 billion. So I don't think, yeah. So if we have our growth, our R is our growth rate. So if our growth rate is 0.5, that means that the population growth rate is increasing. So if R is negative, then that means that more people are dying than are being born. So that would be a negative R. Positive R means that there's more births than deaths. And an R of 0 means that it's just constant. It's nothing's really changing. So if you were asked, does R of 0 interpret a negative growth rate? You would say no. It's just nothing. So it's not positive or negative. Geometric exponential. So you would see an exponential growth curve for an organism that reproduces basically year round. So humans have babies year round. So you would just see like a line. But if you were looking at, say, like a species of plants that only reproduces like during certain months, you would see like geometric reproduction. So you would see like small little dots. You wouldn't see like a straight line. So if you were asked what species would be more likely to reproduce geometrically, it would probably be like the answer would be a species of plants that reproduces at only certain times of the year, right? So this thing is talking about putting a certain bacteria in a jar and seeing what's going to happen. So if we put a population in a jar, it would grow exponentially at first, but then as the population increases too much inside of the jar, there's going to be a depletion of food resources, um, increased aggressive interactions, more waste, more spaced depletion. And then as a result, that's going to lead to slower growth, reduced survival, and reduced fecundity. I wrote that down. Fecundity, the ability to reproduce, the ability to produce an abundance of offspring of new growth. So that's why when you're looking at a population growth curve, a population can only reproduce exponentially until it hits a certain threshold. Because once it hits a certain threshold, there's not going to be enough to basically support it anymore. So you look at, say, like a country like Japan, right? Where I'm pretty sure the population in Japan now actually has a declining growth rate. And that doesn't actually mean that there's anything wrong with the environment. That just means that it hit the carrying capacity and now it's just trying to level out at the carrying capacity. And when a population increases exponentially too much, then that's going to happen. There's going to start to be slower growth, and there's going to start to be reduced survival, more aggression. So if we look at R is our growth rate, right? So with exponential growth, it would just completely ignore the carrying capacity, like humans. K is the carrying capacity. So if we have a species that is able to multiply exponentially, it would just shoot up. It would completely ignore the carrying capacity. Versus logistic growth would kind of grow until it hits the carrying capacity, and then it would even out. So this is known as our K organisms, and this is known as our R organisms, which is something I'll talk about later. That will make sense. Populations of lionfish and other species are limited by negative density dependent factors. These factors, okay, so if we talk about density dependent and density independent factors that limit a population, we're talking about factors that are due to increased population size or factors that have nothing to do with increased population size. So say, for example, if it says the population of a species in an environment reached carrying capacity and now it is unable to grow any further, that would be known as a density dependent factor because it's dependent upon the density of the population. Versus if there's another question that said there was a tropical storm and it ended up killing half of the population, that would be a density independent factor because that tropical storm was completely independent of the population size. It was going to happen anyway. So if you look at a tsunami, a tornado, those are independent, density independent factors because they have nothing to do with population. Some examples of density dependent factors are competition, predation, disease, and social behavior. And that makes sense because obviously if there's a larger population, you're going to see more competition for resources. There's going to be increased predation from other species. There's probably going to be more disease, more parasites, right? All of that makes sense. It's already what I explained. All right. So how do humans get sick? How do pathogens get spread? So another thing that's interesting is humans actually have recently evolved to walk upright, meaning that bipedalism is actually something that's relatively new for humans. I say new, meaning like maybe a couple million years or thousands of years. I don't really know. But in terms of how recent it is for most species, it's fairly recent. So humans aren't necessarily supposed to walk upright. That's why I feel like you see a lot of humans get a lot of issues with their joints as they get older because it's something relatively new that I guess humans have evolved to have. So humans get infected by pathogens. Um, some humans just have alleles that cause them to get sick, like sickle cell disease, right? All right, so this zootonic disease refers to diseases that come from animals. So, say like the COVID or Ebola. A lot of diseases actually come from animals because animals like for example, bats are really good hosts for diseases because if you look at an animal like a bat has a really high metabolism and it has a higher body temperature. So meaning it's able to say have a virus like the coronavirus and not feel sick and live a completely normal life. Meanwhile, it can pass these diseases to humans. So a lot of diseases do come from animals, like HIV came from an animal. So if we look at our different points of transmission, rabies can go from animals to, I think it says only from animals. So rabies only comes from animals versus Ebola can go from an animal to a human, but in um, very limited cycles. It's not extremely contagious. Whereas we look at something like HIV that started from animals into humans, and then it can be passed from humans to humans, like HIV, COVID, and this would be like a disease that you consider probably the most dangerous because it's very contagious and it started in an animal and then it can go from humans to humans rapidly. We use our SIR models to help understand the spread of disease. So our susceptible is the number of susceptible individuals. I equals the number of infected individuals, and R equals our number of recovered individuals. We have our transmission rate, infectious period. This is our curve. So at the beginning, everyone starts as susceptible, and then our oranges are infected. And the SRI model, it's just only assuming that when people get recovered from a disease, they can't get it again, and also that there's no death. So it's kind of like a null model, just like the Hardy Weinberg. But um, you see, everyone starts at susceptible. More and more people end up getting infected. Meanwhile, as people are getting infectable, some people become recovered until basically the end, everyone's recovered, right? And there's no one susceptible or infected. I guess this is kind of a good model to help us estimate how transmission rates and recovery rates can affect each other if we were looking at, I don't know, trying to calculate how long a pandemic would last, possibly. Yeah, so our transmission rate is influenced by the frequency of contact between infected and susceptible individuals and the probability that a person that has the disease will contract it. So also, we're assuming that in this model, there's no deaths, there's no births. We're assuming that infected people can't get infected again. We're assuming that the transmission rate is constant and not changing. And we're assuming that everyone that gets it is immediately um, contagious, right? So it's not the best indication of the real world. Well, that makes sense, but it's just used as a model. Which parameters will we need to change to predict the effects of the following public health interventions? Mask, social distancing, vaccination, testing and isolation. All of this right here is going to influence our transmission rate. So if we increase social distancing, if we made more people wear masks so there was less chance of transmitting a respiratory disease and more people were vaccinated, then all of that would decrease our transmission rate. If that makes sense. And then our food webs. I mean, this makes sense. You learn about the food chain in school. It all makes sense. You have a top predator that eats one thing, that thing eats one thing. It's basically a web. And then if something in the middle goes extinct, it can be harmful for everyone, right? So food webs. All right, this is interactions between two species. So this explains effect on interactions on a certain species, and this affects a certain species interaction on another one. So if two species are competing, it has a negative effect on both species. And then if it has a positive effect on both species, it's mutualism. And then you have somewhere everything in the middle, right? Like we have it where one species benefits and one doesn't get anything in return. That's facilitation. We have one where it's a benefit for one and a negative for another. We get predation, right? So all these organisms interact with each other in very special ways, and they're very, very complex. Like this is a true food web here, and you can just see how many chords there are, and like it's a web, and it's very complex. So if one thing happens to one organism, then it could be dangerous for the entire food web, if that makes sense. Energy is limited in an environment. There are finite energy levels. So, as a general rule of thumb, only 10% of the energy from primary producers gets to herbivores. So, all of this energy gets lost as it goes through the environment. So, there can only be a finite number of energy levels because eventually the energy is just going to run out and there's going to be 0% energy left. And then we have our dominant species, which take up a large proportion of the biomass, and we have a keystone species, which take up a small proportion of the biomass, right? So if we look at a keystone species, we look at a chipmunk. Um, is that a chipmunk? Looks like a chipmunk. A keystone species, it has a big effect on the community despite a lower abundance, a lower biomass. And our dominant species has a large biomass. And if you remove a keystone species, it could be harmful for the environment, obviously, because if you have a brick to a house and you pull out one brick to the house, that might cause the other bricks around it to fall down, and that could make things worse, right? Not all species have a dominant or a keystone species. So if you look at this question here, if you were asked what's a keystone species, we would look at a species that has low biomass but a big impact. So you would see like a sea otter. And if we were looking at dominant species, we would look at, say, trees because they take up a large biomass, right? And this is just another map of how our interactions affect each other. So, for example, species A is preying on species C. This has a positive effect on species A, a negative effect on um, species C, and that's direct. But because species A is preying on species C, this has an indirect positive effect on this has an indirect positive effect on wait no. Let me rephrase what I just said. So species A and species B are competing for resources. Species A preys on species C. So this is direct here. Meanwhile, species C is getting an indirect positive from species B because species B is going after their predator, right? So it's an indirect interaction because if B were to go extinct, then more A would come after C, right? So the species C get an indirect positive benefit slash interaction from the presence of species B. Then we have our trophic levels. We see primary producer, and then we have our herbivores, and we have our carnivore, right? And then once again, we go back to indirect and direct, negative and positive interactions. An island is known as anything as a suitable habitat surrounded by an inhospitable matrix. Larger islands have higher habitat diversity. Larger populations are less likely to go extinct. We already talked about that. Lesson 19, communities. A group of interacting species in the same place at the same time. How do communities form? Um, Evolution, physiological constraints, habitat selection, dispersible ability, species interactions, disturbance and stress, volcanic eruption. Secondary succession is when plants and animals reestablish an area after a disturbance. So I think there was somewhere in Washington, state of Washington, where there was a big volcanic eruption in like 1980, and that just completely devastated the environment. So secondary succession is when plants and animals reestablish an area after a disturbance. So primary succession. So intensity of disturbance. A volcanic eruption would have a high intensity of disturbance, but it happens low. So if we had something that had a high frequency of disturbance and a high intensity, there would be little ability for a population to survive or a community to survive because it would just get nuked too often, basically. But you have something like a tree falling, which has, I don't know if that happens on a low frequency. We look at like waves crashing into a rock, has a very low disturbance, but it happens very, very often. But the thing is, it's actually healthy for an environment to have. Um, it's actually healthy for an environment to have disturbances every so often, right? Kind of like in humans, how there's a healthy amount of level of stress. That's the same way. Like a population needs a healthy amount of stress on the population that is beneficial for the community, right? So I just said secondary selection is when plants and animals reestablish an area after a disturbance. And what's primary succession again? Primary succession is the sequential colonization and growth of bacterial, plant, and animal species in an environment that is previously devoid of life. Okay, so primary succession occurs immediately after the catastrophic event, right? So if we look at an example of a big volcanic eruption, primary succession would be what would need to occur before plants and animals can live in an area, right? That makes sense. Yeah, how does succession work? So this is, say volcanic eruption happens just before here. This would be primary succession, secondary succession that would then become habitable for plants again and animals, right? So our R selected species and our K selected species. I said earlier how K refers to our carrying capacity versus R refers to our growth rate, right? So if we have an R selected species, it means it has exponential growth, it's density independent, it's very good at reproducing very quickly, but it's a bit more unstable. It can die out more quickly. Whereas our K selected species, they're kind of slower at reproducing. Um, they're density dependent, low mortality for offspring, but they're more stable, right? But they tend to hover around a carrying capacity. So if we look at, say, like elephants, right? Like a baby elephant stays with, I think its mom for almost a year, even longer than that. So the offspring are more likely to survive. It's more stable, but they do take longer to reproduce versus something like a plant can grow very, very quickly, but it may not be the best at surviving, right? And it's a bit more unstable. So that's the difference between our R selected species and our K selected species. So yeah, look at this map here. Yeah. Case-selected species in general are just larger organisms, and they tend to live longer and they're more competitive, but also they're slower at reproducing and they're not as good at relocating, right? Like you look at a polar bear. A polar bear is kind of stuck where it is. If there's climate change, it can't really move any farther north. So it doesn't have the best dispersal ability versus I guess like a plant, if it really wanted to, it could just move up a mountain. It would have a higher dispersal ability as a R selected species. Succession occurs via different mechanisms. We have tolerance, inhibition, and facilitation. Facilitation is some species arrive earlier and make it easier for late successional species to invade. Inhibition is some species arrive earlier and make it harder for species to invade later. Intolerance is when it has no interaction. So if we have facilitation, that means that a prior species makes it easier for a new species to live in there, living in the environment. Inhibition means that a prior species makes it more difficult for a new species to move into an environment. And tolerance means that it has no interaction whatsoever. Our intermediate disturbance hypothesis says that if we have an environment that has a really high frequency of disturbance, we're going to see a lot more R selected species because we said earlier that the K selected species are not as good at surviving um, large disturbances. Kind of like 66 million years ago when the meteor hit Mexico and all the dinosaurs died out, yet all the little mice stayed alive. Kind of like that. So, but if we have an environment that has a low disturbance frequency, we would see a higher proportion of K species, right? But if we have something where the frequency is somewhere in the middle, we would see an equal proportion of R and K species. So the question is, at what level of disturbance would K-selected species make up a greater proportion of species diversity? And on the answer here, I would say on the left, because it would have lower disturbance, meaning it would be better for a K-selected species to survive at a lower disturbance level. The intermediate disturbance hypothesis is an ecological concept that suggests species diversity is highest at intermediate levels of disturbance. Kind of what I explained earlier, that it's healthy for an environment to have a specific level of disturbance. Kind of like it's healthy for humans to have a little bit of stress. If that makes sense. It was first proposed by Joseph Cannell in 1978 and provides an explanation for patterns of biodiversity in ecosystems. So I guess this is showing a map of how two different species interact. So our blue is showing an environment where the ova is removed, and this, I think it's called, you called it, Gigartina is alone, right? So in a facilitation model, we would see that when we remove the ova, make sure I interpret this right. Well, you would see in tolerance that they grow at the same proportion. Via an inhibition, we would see that when both of them are present, they grow at a slower rate. Versus in facilitation, when both of them are present, they can grow better together, if that makes sense. So facilitation means that When they're together, they grow better. Inhibition means that when one of them is alone, they grow better. And tolerance is they have no effect of each other whatsoever. So I guess this is a map here where they're removing ova. And as they remove ova, the proportion of gigertina cover increases, which mean that it operates via inhibition because removing one promotes the growth of the other. Yeah, so what process dominate the successional trajectory? Ova is an early successional species that inhibits, however you say that. Ova is more susceptible to environmental stress and consumers. Once ova is removed, the other species dominates. So yeah, it matches well with the inhibition mechanism. So this is referring to the ability of a community to recover after, say, something bad happens to it or a disturbance occurs. Not necessarily something bad, but a disturbance. Resilience, the ability of a community to return back to normal after a disturbance. Resistance of the ability of a community to withstand a disturbance. Return time is the time required to return to equilibrium after a disturbance. So if we have a species that has a low resistance and a low resistance and a low resilience, it's probably eventually going to go extinct because it's only going to take so many disturbances before the community or species just never recovers at all. That makes sense. What happens if a community crosses a threshold? It's basically just going to turn to something else. After crossing a critical disturbance threshold, it will always change and revert to another alternate stable state. Kind of like how in physics class, like, for example, if I were to drop this water bottle, it might bounce on the table once or twice, but then it's eventually going to go back to sitting, right? That's kind of how disturbances work too. It's like I take an environment, I can disturb it, it will eventually get back to a stable level, but it might look a little bit different than it were before. So, for example, this water bottle, I could drop it like this, um, upright, but when it hits, it might bounce over. It might turn sideways. It's still in a stable state. It's still laying on the ground, but now it's just laying sideways. It's kind of different, even though it is at an alternate stable state. The age of dinosaurs spans roughly 186 million years ago. The mass extinction, where the meteor hit Mexico and killed off all the dinosaurs. Oh yeah, I kind of explained to you how when you had a mass extinction event, like here you see the volcano erupt, um, this killed off all the dinosaurs, which actually led to more increased development of mammals, right? So perhaps the extinction of the dinosaurs was what allowed humans to exist. Maybe. It definitely proliferated mammal life because, like I said, species interactions. When that top predator is now gone, that can change an ecosystem. Yeah, adaptive radiation of mammals. Extinction of dinosaurs likely reduced competition for resources among mammals. This ecological opportunity allowed mammals to quickly diversify as they evolved to fill different niches previously occupied by dinosaurs. Makes sense. Adaptive radiation, ecological opportunity makes sense. When you have opportunity, you're more likely to grow. Adaptive radiation occurs when a single lineage produces descendants. Yep, yep, yep. Homo sapiens is the only survivor of an adaptive radiation of hominids that occurred in Africa over the last 4 to 5 million years. That makes sense because you have this common ancestor here, and then it branches off into all these different vast species. And then you look down here, um, we have this common ancestor here that branched into the Neanderthals and the Homo sapiens. And now the Neanderthals are extinct. So Homo sapiens are the only hominid species that are alive after 5 million years. It's quite interesting. So these are the two hypotheses for why that explain bipedalism. So why humans walk upright. I guess the savanna hypothesis thinks that I guess humans adapted towards bipedalism because they transitioned from Africa and they needed to be in more open areas. And the feeding hypothesis says that more upright posture help ancestors feed better in trees, which makes sense. I mean, no one really knows, really. Why is it so difficult to determine what belongs to the genus Homo? Variation within the same species, small sample sizes, potential convergent evolution among hominin lineages. Yeah, so that's another thing that's true. So humans were interbreeding with Neanderthals and other multiple species. So a lot of the DNA is construed, like kind of mixed in. I'm pretty sure modern humans now are like 2% Neanderthal DNA. So it's kind of difficult to determine what group of species belongs to the Homo group because all the species were different. Some of the species were kind of small. Some of the species kind of like reproduce with each other. So it's kind of difficult to understand. So our out of Africa hypothesis seeks to understand why we think modern humans come from Africa. So if we look at here, we have these different species of humans, right? And we have this common ancestor here from tracking the genomes, right? So the most recent common ancestor of all of these groups combined is right here. So the most recent common ancestor is from an African group. So they're thinking that this group here, that's this group of humans is now in Africa. This common ancestor here was also in Africa, and this group is still in Africa, and then that eventually diverged to Europeans, and then Asians, and then Middle Eastern Pacific people. So that's why we think that humans came from Africa because the most recent common ancestor of all humans together is, I guess, African. So that's why they're thinking that humans started in Africa and then moved to other parts of the world because of how the common ancestor works. The out of Africa hypothesis says that Homo sapiens evolved in Africa and moved into Europe and Asia in a second dispersal event. This original hominoid populations did not contribute genes to Homo sapiens during this secondary contact. Yeah, and that's what I said. A lot of these groups were interbreeding as well. So if you look at 50,000 years ago, modern humans were 5% Neanderthals. I mean, obviously, like after thousands of years, when humans are reproducing with other humans, that Neanderthal DNA gets slightly diluted. So now modern humans are only 2% Neanderthal. I mean, that's still kind of crazy that modern humans are 2% Neanderthal. I mean, not like 0.01, like 2%. I mean, that's fairly significant. So that does show that they were interbreeding with other species as well. Ancient humans were interbreeding with other species such as Neanderthals. So, yeah, I put that. If we look here, this shows that once humans basically arrived in a particular area, a lot of organisms went extinct. So, if we look at Africa, hominoids had evolved alongside large animals, so they were more likely resistant to human pressure. So, essentially saying the because of the existence of humans in Africa, 20% of species have gone extinct. And then in Europe, humans arrived 35 to 45,000 years ago, and 36% of the population of all species in Europe went extinct. And then in Australia, humans arrived 40 to 50,000 years ago, and 88% of all of the species went extinct. So that's why a lot of animals today still might be kind of weary of humans because thousands of years ago, a lot of species went extinct due to humans. So that's why some animals become weary of humans, if that makes sense. And then North America, humans arrive 13 to 15,000 years ago. 83% of the population goes extinct. South America, humans arrive 8 to 16,000 years ago, and 72% of the population goes extinct. So humans are predators. A lot of, I think, like billions of species have existed in the world, and because of humans, most of them have gone extinct. That's that's interesting, definitely. Now on lesson 21. As I just mentioned, billions of species have existed, also balanced by billions of extinctions. You see this massive web here. You see how all of these other species have existed and most of them have gone extinct. We have this extinction vortex here. And once a species gets into it, it's very difficult for it to get out of it. So we see once a species gets to a small population size, it's more it's more prone to genetic drift, interbreeding, which lowers genetic variation, could lead to lower fitness, lower ability to adapt, and lower tolerance to disturbances. So you look at something like Neanderthals, right? Like their population gets low, they have to start interbreeding with humans. That ruins their fitness, low genetic variation. It's very hard for them to keep a population up like that. If that makes sense. So eventually, those species will go extinct. I'm not talking about extinctions, right? So we talk about pre-extinction, disaster, recovery, recovered, right? So okay, so we look at R and K selected species. We just said R selected species are can reproduce exponentially versus K selected species cannot reproduce exponentially, but they are more they're they're more competitive, right? And they're usually larger animals, right? So would you expect Bloom taxa to be R or K selected species? We say Bloom taxa would be an R selected species because it can reproduce exponentially with little to no resistance, but it may not be able to keep that up for very long and survive. So we would say that is a answer R selected species. Pre-extinction. So if we look at pre-extinction and after extinction, a lot of the larger animals go extinct, right? So if we look at, say, the dinosaur extinction 66 million years ago, all the dinosaurs, all the large animals go extinct versus most of the smaller organisms are better able to survive that. So smaller species are more likely to survive mass extinctions. Current animal extinction rates within the past 200 years are much higher now than they have been historically, mainly due to humans. I think something like current amphibian extinction rates are 100 times what they normally are. So think of like a frog. Like frog extinction rates are 100 times what they've been historically, primarily due to humans. And that's another reason you got to think like frogs have very permeable skin. So if there's anything like infectious in the water, it's going to impact that. Also, you got to think of like humans building neighborhoods, like all of that impacts biodiversity as well. Caused by climate change, habitat alteration, overexploitation, harvesting, predators and competitors, pollution, and emerging infectious disease. Why is human caused change anything special? Because an individual species has never impacted, a single individual species has never impacted the planet Earth at such a significant rate before as humans have. Like, as one species, humans have done more impact on the Earth than any other species has done ever. That's why human caused change is extremely interesting because it's never quite been seen before in the history of the Earth. And we were talking about NPP and GPP earlier. And we were also talking about how there's a finite number of energy levels. So humans use 30% of the global net primary productivity, meaning that humans are using a significant portion of that energy, meaning that there's less to be passed down to other energy levels. We talked about greenhouse gases. We talked about Milankovitch factors. We talked about why CO2 fluctuates in the northern hemisphere. Why is atmospheric concentration of CO2 rising steadily? More CO2 is being added to the atmosphere each year than is being removed annually. This CO2 is accumulating over time. And I also talked about how the greenhouse gas effect, how solar radiation comes into the earth, it's absorbed by the greenhouse gases, but obviously you have more CO2 in the atmosphere, so more of that is being absorbed, and that's going to increase temperatures. All right, environmental change factors, abiotic and biotic factors. So abiotic factors are non-living factors that can change an environment. So say like weather and geology. So say like abiotic factor would be say earthquake, drought, pollution. You could even go as far as to say an abiotic factor is like rising CO2 levels, because I mean that's not, it's not living. Versus a biotic change is a living organism. So an invasive species, an extinction, disease, right? So how can organisms respond to climate change? Well, individual organisms can adapt, which doesn't actually help the entire species as a whole. It just helps the individual. They can adapt via natural selection and evolution to become more fit for the current environment. They can move or they can go extinct. So if you look at like I mentioned earlier, like polar bears literally cannot go any further north. So if there was a significant increase in Earth's temperature, like a significant increase, they can't really go any further north. There's no way for them to go. So if there is a big change in the environment, individuals have to either adapt, the species has to adapt, or they just have to move or go extinct. Another thing that's interesting that I learned is grizzly bears are moving more north, right? Meaning that grizzly bears are now reproducing with more polar bears, meaning that they're creating something called like pizzly bears, which I guess makes sense. I mean, because that's what happens. I mean, you have species moving and then they'll start to interbreed with each other. And then you get these polar bear, grizzly bear hybrids, which is interesting. Look at that thing. It's like it's brown and white. It's interesting. Okay. What factors limit how a population adapts? How quickly they can reproduce, the genetic diversity, and any isolation or geographical barriers. Like for example, if there's a change in a environment, but the population isn't able to leave, say it's on an island and you're not able to leave, then that species is probably going to go extinct if it's geographically isolated and there's nowhere for it to go. You got to think, like if there's a species of I don't know, a species of dogs on an island, like I don't know how much I trust a dog to cross an entire ocean versus say a bird might be able to migrate versus you have some species that just might not physically be able to migrate to new areas. I already said how far north it already exists. Does a suitable habitat exist? How quickly? I wrote here polar bears are victims of climate change because due to their natural habitat, they cannot go any further north when temperature increases. It's very true. This here is a map that the green dot is historical species richness and the red dot is present species richness. So this is showing that this is altitude. This is showing that compared to historical records, the species richness is higher at higher altitudes. This is meaning that more species are migrating to higher altitudes or up mountains, and that might be due to cooler areas. I mean, species are moving up the mountain range from lower elevation because temperatures up the mountain are cooler. Makes sense. I wrote here, not all species move fast enough to keep up with climate change. Species that can move find themselves without the food they need to survive. This is another thing. If you are a species and you've been living in an environment for, say, thousands of years, and then you move to another environment, there may not be the resources that you need in that new environment to survive. It's very doable. Another thing is leatherback sea turtles. So sea turtles lay eggs on the beach, obviously, and the temperature of the sand determines whether or not the turtle is male or female. So if the if the sand is warmer, that will create a female egg versus if the sand is cooler, that will create a male egg. So if you have, say, a beach and the sand is constantly just getting warmer and warmer, you're just going to have increased number of female sea turtles. So either what's going to happen is, I guess they're just going to have to start laying eggs earlier in the cooler months, or there's just going to be a lot more females than males, and that definitely isn't good for a population reproduction wise. Conservation biology embraces certain values. Yep. Conservation biology is essential for addressing the biodiversity crisis and ensuring the health of ecosystems that humans and all life forms depend on. It seeks not just to preserve what exists today, but to maintain the processes that sustain life for future generations. Investigate human impact on genetic variation, species, and ecosystems. How can it be practiced? How can we be practical? Develop approaches that prevent species extinction, maintain genetic diversity, and protect biological communities and their associated function. Cool. Yeah, but this is how all these how medicine interacts with conservation biology. That makes sense. Like medicine, you need to understand A&P, biochem, genetics, conservation biology. You need to know evolution, ecology, genetics, forestry. Goal is to protect species. Goal of medicine is to improve human health. How do we measure success in conservation biology? Did we prevent extinction? Have we maintained or increased genetic diversity? Are biological communities and their associated functions persisting through time? That's a good measurement. If I'm thinking, okay, I want to try to conserve biology, I feel like the question would be, can I maintain the ecosystem or improve it as it currently is? Can I prevent the extinction? And how can I help that community persist and stay healthy? That makes sense. Extinction is very rapid. Speciation is slow. The future is unknown. All right. I think that covers all the lecture parts. This is just a list of learning objectives that I answered. I can go through some of them. I mean, I literally throughout the course of the nearly 2 hours I've been talking, I've pretty much gone over all of these. So I'll answer these: independent, dependent variable, testable hypothesis, Shannon Weiner, alpha, beta, gamma diversity, reflection plots, energy in an ecosystem, CO2 rates, climate change, nitrogen cycle, evolution, allele frequency, Hardy-Weinberg, genotype frequencies, reproductive competition, conservation biology, food webs, keystone and dominant species, predators. Oh, immigration curves. I need to talk about immigration curves. I think that, where was that at? That was exam 3. I need to talk about, yeah. So we haven't talked about immigration yet. All this I've already gone through. Oh, herd immunity. Herd immunity is basically saying like once you get a significant proportion of a population like vaccinated, that then significantly decreases the probability of it spreading throughout the entire population. If that makes sense. Food webs, trophic levels, interactions, climate change. All right. Immigration. Rank these species in order from highest species richness to lowest species richness. Well, A is going to have the highest species richness because we're looking at and island size and island proximity. So A is going to have the biggest species richness because it's closer to mainland. D is going to have the second biggest species richness because it's closer to mainland. C is going to be third because it's the biggest, even though it's a bit farther. And B is going to have the lowest species richness. Larger islands have higher habitat diversity. Large islands have larger population sizes, so typically lower extinction rates. Large islands are usually closer to mainland. Oh, this is a question. Yeah, A and B would be extinct because large islands are not necessarily closer to the mainland. That island size and creation is pretty random, but A and B is correct. So the answer is B. You would typically think a larger island would have more habitats and a larger population is less likely to go extinct, right? Islands near a continental source may have higher immigration rates. Assumptions. Species richness results from a dynamic balance between immigration and extinction processes. Okay. Immigration rate. The number of species arriving to the island over time, highest when the island is unoccupied. Extinction rate. The number of species that go extinct on the island over time, highest where species number are at a maximum. Yeah, so smaller islands should have higher extinction rates because you have to think if you have a smaller island, that typically means a smaller population more likely to go extinct. Isolations closer to the mainland are going to have higher immigration rates because obviously there's going to be that closer proximity. So there's going to be more like it's going to be easier for a species to travel. So smaller islands have higher extinction. Islands closer to the mainland, higher immigration. So these are different immigration and extinction curves. We want to determine what it is for each island. So, which letters represent the immigration and extinction curves of species richness of B? So, B, it's going to be is farther away. So, it's going to have high extinction. It's going to have high extinction but low immigration. So, if we're looking at okay, so I'm pretty sure which letters represent the immigration and extinction curves of island B. So it's going to be B, which is X, Y, S1. So it is B because it will have a low immigration curve because it is far away. So I guess it doesn't really tell you which one is immigrant. I mean, that would be helpful. But it's gonna be this one, S1 here, because it's gonna have that low immigration but high extinction. So it's gonna be that extreme there. So it will have a low immigration curve because it is far away. It will have a high extinction rate, Y, because it is a small island. It is S1 because it will have the lowest number of species at equilibrium. Yeah, so S1, S2, S3, S4 refers to when the extinction and the immigration rates are at equilibrium, what would have the highest species. So we know that A would be the one that will be able to have the most amount of species. So we automatically know that S4 is A because it's going to have high immigration, low extinction, right? Which island has the highest rate of species turnover at equilibrium? We would say D, because we're looking at what would be S3, because S3 is going to have this S3 is equilibrium, right? And this is going to be the highest turnover rate. And it's D because D is a smaller island, so it's going to have higher extinction, but also higher immigration. So it's going to meet here at S3, which would be the highest turnover rate at equilibrium. So I said D equals WYS3, which is the highest point. That makes sense. Which island will have a greater number of species after 1 year? Near or far island? Near island, because it's going to be closer. Why aren't lichens and tigers and bears found everywhere? Continents move. Okay, this is talking about drift. Okay. Why aren't lions and tigers and bears found everywhere? That's because continents move over long periods of time due to tectonic activity, right? Yeah, so 60 million years ago, South America spent, yeah, it was an isolated island. The Great American Biotic Exchange. Um, yeah, North America, elephants, camels, dogs, and cats. All of these animals came from North America. Then from South America, that came from South America. All right, Species richness is higher at the equator because it has the highest NPP. That makes sense. We talk about NPP and GPP. We talk about how there's a finite amount of energy, right? So if there's obviously more energy available in an environment, then that means that there's going to be ability to maintain more energy levels. So if it was asked what level of the Earth has the highest species diversity or richness, you would say the level with the highest NPP. That makes sense, right? Because there's the most energy there to maintain the most amount of life. Why are there more species in the tropics? If you look at Antarctica, there's barely any species. Closer to the North Pole, there's barely any species. What makes the tropics different? Moderate temperatures and plenty of sunlight, larger area of land mass, no glaciers during ice age. Bunch of factors. So I guess this image here, the water droplet represents a species, and the bucket, I guess, um, represents the ability to maintain life. So if you look at a tropic zone, the bucket's bigger because it has more energy to support life. Versus a more temperate zone, the bucket's smaller because it can't support much life. And I guess evolution at tropic regions happens quicker, and it has a longer history too because, like I said, there's no ice ages just stopping everything every once in a while, if that makes sense. All right, multiple different hypotheses have been proposed to explain the lateral diversity gradient of the tropics. Data in Graph C supports the hypothesis that the tropics are more diverse because of higher productivity and greater resource availability, therefore, more species can coexist. Which of the remaining two graphs have data supporting the hypothesis that the rate of speciation in the tropics is greater than in temperate regions? Select your answer. Well, graph B is just showing that they start at different times. So that doesn't necessarily prove that the rate of speciation in the tropics is greater. I think I think C would represent like ecological limits because it looks like in the temperate region it's like kind of plateaued to what its um, species richness is. But graph A has shown that over the same amount of time that the tropics has been able to increase its species richness, right? Yeah, because which hypothesis supports the rate of speciation in the tropics is greater than in temperate regions? That would make sense. It would be graph A. More learning objectives, more learning objectives. Factors affecting disease spreads, population density, transmission rate, infectious period. Exam review, exam review. Yeah, like I said, graph C represents the productivity of an environment. History represents how long it's been happening. For example, like if every thousand number of years you get a random ice age that stops all of your processes, then of course you're going to have split up times. But this one here, graph A, shows that evolutionary rates and speciation is higher in the tropics than in temperate regions. Practice questions, practice questions. Which biological rate from the equilibrium theory of island will be affected by shrinking forest habitats? So when a habitat shrinks, the extinction rate will increase. And then by shrinking, it will become further away from the larger mainland. So the immigration rates will decrease. Pretty straightforward. Number 20, the California condor is the largest North American bird. It's endangered. In 2020, there was a wildfire and they were displaced. 11 had died. By burning such a large area of land and killing some condors, did the fire exert a density dependent or density independent population? Answer briefly. It would be density independent because that fire would have affected the population regardless of how big it is. It would have happened regardless of how big the population size was. So it was density independent. Okay, so that sums up immigration. Immigration curve, species richness. Primary succession occurs on barren land with no soil, while secondary succession occurs in areas where soil remains after disturbance. So yeah, primary succession occurs like right after, like immediately after the disturbance, versus secondary is after that. R and K species, intermediate disturbance hypothesis. Resistance, resilience. Morphological innovations like bipedalism and larger brain sizes allowed early humans to exploit new ecological niches, enhancing survival and reproduction. Out of Africa hypothesis, interment phylogenies, conservation biology, NPP, GPP, rising CO2, conservation biology. All right. So that goes through all of the main learning objectives. And I went through exam 3 as well. This is just going basically over the other YouTube video I did, explaining everything I've already talked about. This was for the Unit 1 test. These were exam practice questions that I did. This was another file that I had for exam 2. Figure I'd look over that, make sure I got everything that I wanted to talk about. Mutations. Okay, so apparently there was a dentist that had HIV that was practicing without gloves in like the 90s, and they were trying to figure out the phylogeny to figure out, was the dentist giving the patients HIV. So this is the clade that would basically say that the dentist was, or the phylogeny that would say that the dentist was giving the patients HIV, because this is the control, which is just civilians that have HIV. And this is HIV, and this is the dentist, and he's giving it to the patients. You can see right here. And this chart here is the dentist did not infect the patients. As you can see, the only people getting infected with HIV are the ones associating with the people that already have HIV, if that makes sense. So that'd be how you could read a clade or a phylogeny and see if something happened. In small populations, rapid changes in allele frequency are totally independent of mutation and natural selection. Yeah. So say if you have two alleles in a population, over many number of generations, one of those alleles are going to kind of fade out and one allele is going to kind of dominate, So if you have allele big A and allele little a, over time, one of those alleles is going to fade out over a great number of generations. So you look here, generation zero, the allele frequencies is zero point 0.5, everything's equal. After one generation, it kind of changes. And after generation 16, you have some groups that'll be 100% like big allele A, and you'll have another group that'll be 100% little allele A. So that's just how genetic drift works over time. Natural selection, mutation, migration, genetic drift, non-random mating, monophyletic group, paraphyletic group, polythetic group. Some more notes. Male-to-male competition, phylogenetics, math, more review, more review, more review, lesson objectives, cladograms. This is a practice test. Sam becomes ill with a UTI. Sam's doctor prescribes an antibiotic for this infection. A few days after taking the antibiotic, Sam feels better. Then he stops taking the pills, even though some pills remain. Shortly, his symptoms return. So this means that the UTI infection has now mutated to become resistant to the medication that he takes. So a drug-resistant mutation existed in the population of bacteria prior to the drug entering Sam's system, stopping the antibiotics, allowing the resistant. Yeah, that makes sense. Yeah, so it's C, not A, because the bacteria wouldn't have mutated as a result of the medication. There was other bacteria that was resistant to the antibiotics that he or she was taking. So that makes sense. Hardy-Weinberg. We use the Hardy-Weinberg model to determine if a population is experiencing evolution and therefore apply several assumptions to mathematical model. Which of the following would be true for the squirrel population if it does meet the Hardy-Weinberg assumptions? So remember, the Hardy-Weinberg is a null model. So the squirrels would have to mate randomly, and the population would have to be infinitely large. Because remember, in Hardy-Weinberg, there's no migration, and there will be no mutations. So A and B are correct. If mutations arrive spontaneously, the will arise before. I need to review that image there. So Lauren, their class experiment. If mutations arrive spontaneously, meaning I guess random, Yeah, that one I need to go back and look at. I think I have a picture of it somewhere. Yep, natural selection, evolution. Okay. I'm going to go back and see if I have an explanation for this one. Because I believe I did talk about it. It was from all the way back. It was from the first exam. Hmm. I have to find an explanation for that, but I'll do that lastly. I'll go ahead and continue going through all of these. There's just more practice questions, practice questions. I'm going through all of this fairly quickly because I have to leave soon, but I've talked about most of this already. Genotype, frequency, more practice questions. Talked about most of this. More learning objectives, more exam review, more questions. Okay, what was this one? Yeah, this is the one that I just came across. I'm going to find the answer for that somewhere. I can't go through all the questions right now because I'm on limited time. I believe I have talked about everything. I will find the answer to that question. Give me 1 second. Sure of. This is saying like if mutations arrive naturally, resistant cells will arise before exposure to a phage, which that would make sense because it would just be random. The resistant mutations occur late during the population growth and result in few resistant cells, but occasionally resistant mutations occur, resulting in many resistant cells. As a result, variance in the numbers of resistant cells among replicant runs is expected to be high. And then, yeah, a random mutation, a resistance mutation occurs after the phage is present. Every cell will not have an equal probability of mutating to resistance. Induced mutation, resistance mutations occur before the phage is present. Every cell will have an equal probability of mutating to resistance. Interesting. So, as for pretty much covered the entire course, I mean, there are a couple of practice questions that I have to go through myself um, because I have to go take a test shortly. But I think I taught the entire class. So yeah, I think I taught the entire class. Definitely helped me review a couple hours that I did this. Um, I hope I helped any of you that are maybe doing this class. I know I primarily do this for my own studying as well. So